Microbiology Chapter 6, Acellular Pathogens. Public health measures in the developed world have significantly reduced mortality rates from viral epidemics. However, the rapid spread of these epidemics can still occur due to global air travel. For instance, in 2009, the H1N1 influenza outbreak quickly spread across multiple continents. Similarly, in early 2014, an Ebola outbreak in Guinea escalated into a major epidemic in Western Africa, leading to concerns when infected individuals traveled to the United States, raising fears of a wider spread beyond Africa. The COVID-19 pandemic requires no introduction and is considered the deadliest since the 2018 Spanish flu. So the discussion on virology has come into a very household since then, and the stakes for pandemic preparedness cannot be higher. Before the late 1930s and the invention of the electron microscope, viruses were unseen to the human eye, yet methods for the prevention and treatment of viral infections had already been developed. Historical records indicate that inoculation or variolation was practiced as early as 17th century to combat smallpox in various regions. By the late 18th century, Edward Jenner, an Englishman, pioneered vaccination by inoculating individuals with cowpox to prevent smallpox, marking a significant advancement in immunization techniques. In modern times, while the structure and genetics of viruses are well understood, ongoing research continues to uncover their complexities. This chapter will explore the structure, classification, and cultivation of viruses, as well as their effects on hosts. Additionally, we will examine other infectious agents, such as viroids and prions, to gain broader understanding of acellular pathogen infections and their implications. In 2014, Ebola epidemic in Western Africa was one of the most severe outbreaks of Ebola virus in history, primarily affecting countries such as Guinea, Liberia, and Sierra Leone. Beginning in late 2013, the epidemic quickly escalated with thousands of cases reported and a high mortality rate, estimated at around 40%. The outbreak strained the healthcare systems, overwhelmed medical facilities, and led to widespread fear and stigma surrounding the virus. International response efforts included the deployment of medical teams, establishment of treatment centers, and public health campaigns aimed at educating communities about prevention and control measures. Despite these efforts, the epidemic highlighted significant gaps in global health infrastructure and need for improved readiness to tackle infectious disease outbreaks in the future. The outbreak started in late 2013, but was only confirmed months later in 2014. It spread from Guinea to Liberia, Sierra Leone, Mali, Senegal, and Nigeria. After 28 months, the outbreak had resulted 28,652 cases and 11,325 deaths. Of the dead, 518, about 5%, were healthcare workers. In response to the Ebola outbreak, the much-awaited viral vector Ebola vaccine, RSV-ZBOV, also known as a ZMapp, was approved by FDA in 2019 in US and Europe, created by scientists at the National Microbiology Laboratory in Winnipeg, Manitoba, Canada, which is part of the Public Health Agency of Canada, PHAC. PHAC licensed it to a small company, Newlink Genetics, which started developing the vaccine. Newlink in turn licensed it to Merck in 2014. It was used in the Democratic Republic of Congo in a 2018 outbreak in Equatorial Province, and has since been used extensively in the 2018-20 Kivu Ebola outbreak with over 90,000 people vaccinated. The COVID-19 pandemic caused by the novel coronavirus SARS-CoV-2 emerged in the late 2019 and rapidly spread worldwide, leading to unprecedented health, social, and economic challenges. First identified in Wuhan, China, the virus quickly crossed borders, resulting in millions of infections and deaths globally. Governments implemented various measures, such as lockdowns, social distancing, and mask mandates to mitigate transmissions, while healthcare systems faced immense pressure. The pandemic highlighted disparities in healthcare access and the importance of public health infrastructure. Vaccination campaigns rolled out in late 2020, providing hope for containment and recovery. The global response to COVID-19 emphasized the need of international collaboration in addressing public health threats and underscored the critical role of science and research in developing effective vaccines and treatments. The evolutionary origin of SARS-CoV-2 remains controversial. However, scientists believe that the virus possibly originated in bats before being transmitted to an intermediate host, likely a pangolin, never confirmed, where it mutated and then transferred to humans. Most viral evolution occurs by gradual stochastic accumulation of substitutions over lineage generations in adapting host environments that might ignore population heterogeneity. Alternatively, viral evolution can be caused by a cluster of mutation that appear simultaneously, resulting in sudden jump in viral evolution. In the COVID-19 pandemic, variants of concern VOC variations in alpha, beta, gamma, delta, and omicron show extensive genomic mutations from the original Wuhan 01 strain, with enhanced viral infectivity and immune jeopardizing in humans. To date, Omicron is the most divergent VOC or strain that spreads unpredictably fast and crafty, which has completed the evolutionary jump by infecting a large population of human hosts as the Delta outbreak in India in 2021. Although viruses constantly evolve and mutate, it is worth noting that the mutations do not necessarily mean that a virus becomes more dangerous or transmissible. Therefore, it is valuable to understand the mutation or evolutionary patterns based on the big data of SARS-CoV-2 for further developments of vaccines and treatments for COVID-19. So, what is a virus anyway? In 1892, the discovery of a filtrable component smaller than the bacterium that causes tobacco mosaic virus emerged, despite its small size making it invisible to light microscopes. Russian botanist Dmitry Ivanovsky identified the source of TMD by utilizing a porcelain filtering device invented by Charles Chamberlain and Louis Pasteur in 1884. These Chamberlain filters, with a pore size of 0.1 micrometer, effectively removed all bacteria measuring about 0.2 micrometer from the liquid samples. Ivanovsky extracted material from TMD-infected tobacco plant to investigate the disease cause, initially suspecting a bacterial origin. However, he was surprised to find that the extract still contained the causative agent after passing through the filter. This led him to hypothesize that the cause of TMD could be an extremely small bacterium or bacterial spore. Other scientists, including Martinus Beijerinck, further explored the cause of TMD, with Beijerinck concluding in 1899 that the agent was not a bacterium, but instead a chemical, potentially a toxin. Consequently, the term virus, derived from the Latin meaning poison, was adopted to describe the cause of TMD a few years after Ivanovsky's discovery. Although Ivanovsky never could visualize the virus and mistakenly believed it to be a bacterium, he is recognized as the original discoverer of viruses and a founding figure in the field of virology. Viruses, observed through electron microscopes, are unique biological entities whose evolutionary origins remain speculative. They are not part of the tree of life due to their acellular nature and classified as obligate intracellular parasites since they require a cellular host to survive and reproduce. When a virus infects a host cell, its genome directs the production of essential viral components, including protein and nucleic acid, to create new virus particles known as virions. These newly formed virions then carry the viral genome to infect additional host cells, facilitating further rounds of infection. Definition of a virus: an infectious, obligate, intracellular parasite comprising genetic material, DNA or RNA, surrounded by a protein coat called capsid, sometimes a membrane, phospholipid biological membrane. This is probably an accurate definition for 99.9% of the viruses, except beneficial viruses or commensal, which neither benefit nor harm host viruses. Infectious means it can go from cell to cell or host to host. It can go into a cell, replicate, infect more neighboring cells, or transmit to other hosts or individuals. Parasite means it depends on the host to survive, kind of benefit itself, and harm the host somehow. Now, obligate intracellular parasite means every virus needs a living host cell, be it animal, plant, insect, or fungi or bacteria, to replicate, reproduce, or propagate. Obligate means they are bound to or have to follow this rule without exception. For example, the virus can be present in the blood but can't replicate. A virus needs to enter a blood cell, say a CD4 T cell for HIV, to replicate. Unlike some bacteria which can replicate simply in the blood, because a bacteria is not an obligate intracellular parasite. A bacteria may be an obligatory parasite, which means it may need a host to survive, which means it may need at least a host tissue fluid to survive, but does not require a cell to survive. Then the huge difference from any living organism is that some viruses are RNA-based. There is no living system on the planet with an RNA genome. Apparently, there is a limitation on the size of the RNA genome. Beyond that limitation, they become unstable and prone to degradation. Environmental factors can easily destroy them. But DNA genomes are very stable in contrast. Think about a virus as if it is a box containing a present. The present itself inside the box is the DNA or RNA genome or the genetic material. The box protects this present, say cardboard, just it helps to protect the present in a shipment. This box of a virus is made of a protein coat. Now, sometimes the box with the present can be wrapped with a wrapping paper. The wrapping paper surrounding the box is analogous to phospholipid membrane that sometimes surround the protein coat. Now, sometimes you decorate the box wrapper as well as with stickers and bows, etc. The same way the virus decorates this membrane with viral proteins. This membrane is called an envelope of the virus. Remember, not all viruses have that. The viruses without it are called the naked viruses, and with it are called the enveloped viruses. All coronaviruses are enveloped viruses, and the spike protein that you hear all the time is a viral attachment protein, just like a decorative bow on the box wrapper. There are two other proteins in addition to the spike proteins, as well as envelope E protein and membrane M protein, as shown in the C figure of the slide. Virions or viral particles are notably small and cannot be seen with standard light microscopes. As they are significantly smaller than both prokaryotic and eukaryotic cells, which enables their ability to infect these larger cells. Their sizes typically range from 20 nanometer for small viruses to 900 nanometer for larger ones, but recent findings have uncovered giant viruses like Pandoravirus salinus or Pithovirus sibericum, which are nearly the size of a bacterial cell. This slide actually shows a relative comparison of the viruses and other living and non-living entities. In 1935, Wendell Stanley became the first scientist to crystallize the structure of a tobacco mosaic virus using electron microscope, revealing that it is made up of RNA and protein. He later isolated the influenza B virus in 1943, aiding in the development of a flu vaccine. Stanley's groundbreaking discoveries clarified the nature of viruses that had baffled scientists for over 4 decades, earning him the Nobel Prize in 1946 for his significant contribution to virology. Viruses are complex entities consisting of a nucleic acid, either RNA or DNA, encased in a protein coat known as capsid. Unlike a cell, the interior of the capsid does not contain cytosol. Instead, it holds essential components required for virus's replication, including the genome and specific enzyme. The capsid itself is made up of a protein subunit called capsomeres, which can be composed of one or more types of capsomere proteins that fit together to create a tightly packed structure. Viruses can be classified into two main categories based on their structure: naked or non-enveloped viruses, which consist solely of nucleic acid and a capsid, and enveloped virus, which have nucleic acid packaged in a capsid surrounded by a lipid bilayer. This lipid membrane, known as the viral envelope, is derived from the host cell as the virion exits. The origin of the viral envelope can either be internal or cytoplasmic, reflecting its method of acquisition during the budding from the cell. Some viruses, both naked and enveloped, possesses protein structure called spikes that extend from the capsid. These spikes play a crucial role in the virus's ability to attach to and enter host cells. For instance, in influenza virus, specific spikes such as hemagglutinin H and neuraminidase N facilitate attachment and release from the host cells, respectively. Influenza viruses are often recognized based on these spikes, leading to designations like H1N1, H2N2, H3N2, and so on, which have been associated with significant pandemic throughout history. Viruses exhibit a variety of capsid shapes, which can be classified as helical, polyhedral, or aka icosahedral, or complex. Helical capsid found in viruses like in the naked tobacco mosaic virus and the enveloped Ebola virus are cylindrical or rod-shaped, housing the genome within their length. In contrast, polyhedral capsids such as those in poliovirus and rhinovirus are characterized by their icosahedral structure. Both helical and polyhedral viruses may possess envelopes. Complex viral shapes seen in certain bacteriophages like the T4 phage and pox viruses such as the vaccinia virus incorporate features of both polyhedral and helical forms. In these complex structures, the genome is contained within a polyhedral head connected to a sheath and links to tail fibers and pins, allowing the virus to attach to the host cell receptors. Poxvirus with complex shape often display a brick-like form and intricate surface characteristic not found in the capsid categories. This slide shows the 5 structural forms of viruses in nature. They are naked icosahedral, example poliovirus, adenovirus, hepatitis A, rhinovirus, naked helical, tobacco mosaic viruses. So far, no animal viruses of these structures are known. The enveloped icosahedral, example herpes virus, yellow fever, rubella. Enveloped helical, rabies, influenza, mumps, measles virus. Enveloped helical, rabies virus, influenza virus, mumps, measles. Complex are the pox virus, Pandora virus, Mimi virus, many bacteriophages, but not all. The helical symmetry as an example. The nucleocapsids of some enveloped animal viruses and certain plant viruses and bacteriophages are rod-like or filamentous structure with helical symmetry. Helical symmetry is described by the number of structural unit per turn of the helix screw, mu, the axial rise per turn, rho, and the pitch of the helix, p, is given by the formula p is equal to rho multiplied by mu. A characteristic feature of the helical structure is that any volume can be enclosed simply by varying the length of the helix, such as the structure is said to be open. In contrast, capsid with icosahedral symmetry, going to discuss, are closed structures with fixed internal volume. From structural point of view, the best understood helical nucleocapsid is the tobacco mosaic virus, the very first virus to be identified. The virus particle comprises of a single molecule of plus-strand RNA, about 6,400 bases in length, enclosed within a helical protein coat. The coat is built from a single protein, so for TMV, the subunit and the structural units are the same. That folds into an extended structure shape like a Dutch clock. Repetitive interactions among coat protein subunits form discs that have been likened to clock washers, which in turn assemble as long, rod-like right-handed helix. In the interior of the helix, each coat protein molecule binds 3 nucleotides of the RNA genome. Therefore, the coat protein subunits engage in identical interactions with one another and with the genome, allowing the construction of a large stable structural form, multiple copies of a single protein. In TMV, there are 16.3 protein subunits per turn, and each protein subunit has a 1.4 angstrom row or axial rise. So the pitch of the helix is approximately 22.82 angstrom. Calculation of P is equal to rho multiplied by mu. For TMV, it's 16.3 multiplied by 1.4, that is 22.82. So the pitch of the helix is approximately 22.82 angstrom. And below is the calculation details. An icosahedron is a solid with 20 triangular faces, 30 sides, and 12 vertices related by 2, 3, and 5-fold axis of rotational symmetry. In a few cases, the virus particles can be readily seen to be icosahedral. However, most closed capsid look spherical, and they often possess prominent surface structure or viral glycoproteins in the envelope that do not conform to the underlying icosahedral symmetry of the capsid shell. Nevertheless, the symmetry with which the structural units interact is that of an icosahedron. Viruses, though not classified within the 3 domains of life, require a classification system due to their vast numbers. Since 1971, the International Committee of Taxonomy of Viruses (ICTV), assigned by the International Union of Microbiological Societies Virology Division, has developed a universal virus taxonomy. Due to a rapid mutation of the viruses, classifying them using traditional binomial nomenclature is challenging. Instead, the ICTV categorizes the viruses into families and genera based on their genetics, chemistry, and morphology, and replication mechanisms. Virus taxonomy has evolved since its inception in the mid-20th century, with a traditional focus on closely related viruses. Recently, the International Committee of Taxonomy of Viruses has expanded its classification framework to include basal evolutionary relationship among distantly related viruses, adopting a 15-rank classification hierarchy that aligns with the Linnean system. This change allows for a more comprehensive representation of genetic diversity in the virosphere. This 15-rank classification has classified viruses into 14 orders, 150 families, and 1,090 genera, with family names ending in viridae, example parvoviridae. The genus names in virus, example parvovirus, all of which are italicized. Viral species are often referred to using a genus and a species epithet, such as Pandoravirus dulcis or Pandoravirus sellinus. Because the viral genome carries the entire blueprint for virus propagation, molecular virologists have long considered it the most important characteristic for classification purposes. Therefore, although individual virus families are known by their classical designation, they are more commonly placed in groups according to their genome types. There are 7 genome types for all families of viruses. As you know, that Francis Crick conceptualized the central dogma of molecular biology for flow of genetic information, DNA to RNA to protein. This could be extended to the viruses as well, with little modification, DNA or RNA to RNA and then protein. All viruses must direct the synthesis of mRNA that is decoded by the host translational machinery. Appreciation of the essential role of the translational machinery inspired David Baltimore to devise a complementary classification system. This scheme describes the pathways for the formation of mRNA for viruses with either DNA or RNA genomes. By convention, mRNA is defined as a positive plus strand because it contains immediately translatable information. In Baltimore's classification, a strand of DNA that is equivalent to a sequence is also designated as a plus strand. The RNA-DNA complements of the plus strands are designated negative strands. The information embodied in this classification provides the virologist with immediate insights into the steps that must take place to initiate replication and expression of the viral genome. This slide shows the classification and taxonomy of the viruses based on their genome, double-stranded DNA enveloped, double-stranded DNA naked genome, single-stranded DNA naked, double-stranded RNA naked, plus single-strand RNA naked, plus a single-stranded RNA enveloped, and minus single-stranded RNA enveloped. So what is the difference between the naked and the enveloped? We already talked about that enveloped virus do have a membrane outside its capsid. In this section, we will cover several key aspects of viral biology, including the lytic and lysogenic life cycles of bacterial viruses, which describe how viruses infect and replicate within host cells. We will also discuss the replication processes specific to animal viruses following the Baltimore scheme, detailing the unique characteristic of retroviruses and latent viruses. Additionally, it discusses human viruses and their interaction with host cells, explains the process of transduction, which is the transfer of genetic material between bacteria via viruses, and describes the replication process of plant viruses. Viruses rely on a host cell for reproduction and metabolic function as they lack the necessary enzyme for replication on their own. Once inside a host cell, viruses can utilize the cell's machinery to create more viral particles. Bacteriophages specifically replicate in the cytoplasm of prokaryotic cell, while most DNA viruses in eukaryotic cells replicate within the nucleus, except for large DNA viruses like pox viruses that replicate in cytoplasm. Additionally, RNA viruses that infect animal cells typically replicate in the cytoplasm. The life cycle of bacteriophages serves as a valuable model for understanding virus behavior in infected cells, reflecting processes seen in eukaryotic viruses that can either kill the host cell outright or lead to persistent infections. Virulent phages usually result in cell death through lysis, while temperate phages can integrate their genomes into host chromosome, replicating alongside with the host genome until triggered to produce new viruses. Lytic cycle. During the lytic cycle of virulent phages, the bacteriophage seizes control of the bacterial cell, produce new phages, and ultimately destroy the host cells. T-even phage exemplifies a well-studied group of virulent phages. The lytic cycle consists of 5 stages as shown in this slide figure. The first stage, attachment, involves the phage binding to specific receptors on the bacterial surface, such as lipopolysaccharide or OMPC, which is an outer membrane protein C. It is a porine protein. Most phages have limited host range, targeting a specific species or strain of bacteria, which can be utilized in the phage therapy for treating bacterial infection. This could be a next big thing in antibacterial pharma and biotech industry, or in phage typing to identify distinct bacterial subspecies. The second stage, entry or penetration, occurs when the tail sheath contracts, functioning like an hypodermic needle to inject the viral genome into the bacterial cell through its wall and membrane, while the phage head and the other components remain. The third stage of infection involves biosynthesis of new viral components. Once the virus enters into the host cell, it produces virus-specific endonucleases that break down the bacterial chromosome. The virus then takes control of the host cell's machinery to replicate, transcribe, and translate essential viral components, including capsomeres, sheath, and base plates, tail fibers, and viral enzymes needed for assembling new viruses. RNA polymerase genes are typically activated early in this cycle to produce mRNA, followed by protein synthesis and replication to form genomic RNA and viral structural proteins. The first event usually is the early mRNA synthesis, followed by early non-structural protein translation. These non-structural proteins, not found in the viral structure but only in an infected cell, seize control of the host cell and also are enzymatic components for the next step of biosynthesis. In this next step, the non-structural proteins help in the full-length genome replication, which will be packaged in newly formed virions. In addition, late mRNAs are also translated into late structural proteins, which are capsomeres, sheath, baseplate, tail fibers, etc. During the maturation or assembly phase, the new virions are formed. To release free phages, the bacterial cell wall is broken down by phage proteins like choline or lysozyme. The final stage is the release of the mature viruses, which burst out from the host cell in a process known as lysis, allowing the progeny viruses to be released into the environment to infect new additional cells. In a lysogenic cycle, the phage genome enters into the host cell through attachment and penetration. A well-known example is the lambda phage. During this cycle, the phage genome integrates into the bacterial chromosome instead of destroying the host, becoming a part of it known as prophage. The bacterium harboring this prophage is referred to as a lysogen. The infection of a bacterium by a temperate phage is known as lysogeny, where these phages typically remain inactive within the cell. As the bacterium duplicates its chromosome, it also replicates the phage DNA, passing it to the daughter cells during reproduction. This presence can change the bacterium's phenotype by introducing new genes, such as toxin genes that can enhance virulence, a common phenomenon called lysogenic conversion or phage conversion. For instance, bacteria like Vibrio cholerae and Clostridium botulinum are less virulent without their prophage, as the phages carry the toxin genes that boost host virulence when expressed. In Vibrio cholerae, the toxin can result in severe diarrhea, while in Clostridium botulinum it can lead to paralysis. The prophage remains within the host chromosome until an induction event occurs, leading to the excision of the viral genome. After induction, the temperate phage can enter into a lytic cycle and potentially undergo lysogeny in a new host cell. Transduction is the process by which DNA is transferred from one bacterium to another by a virus. It also refers to the process where a foreign DNA is introduced into another cell via viral vector. Transduction does not require physical contact between the cell donating the DNA and the cell receiving the DNA, which occurs in conjugation, and it is DNase resistance, whereas transformation is susceptible to DNases. Transduction is a common tool used by molecular biologists to stably introduce a foreign gene into a host cell genome. Transduction happens through either the lytic cycle or the lysogenic cycle. When bacteriophages, viruses that infect the bacteria that are lytic, infect the bacterial cell, they harness the replicational, transcriptional, and translational machinery of the host bacterial cell to make new viral particles or virions. The new phage particles are then released by the lysis of the host. Generalized transduction. Generalized transduction is a rare event that occurs on the order of one phage in 11,000. Generalized transduction occurs when a random piece of bacterial DNA are packaged into a phage. It happens when a phage is in the lytic stage. At this moment, that the viral DNA is packaged into a phage head. If the virus replicates using head-full packaging, it attempts to fill the head with genetic material. If the viral genome results in spare capacity, the viral packaging mechanism may incorporate bacterial genetic material into the new virion. Alternatively, generalized transduction may occur via recombination when the bacterial DNA packaged into the virus is inserted into the recipient cell. These things can happen. DNA is recycled for spare parts. If the DNA was originally a plasmid, it will recircularize inside the new host cell, become plasmid again. If the new DNA matches with an homologous region of the recipient cell's chromosome, it will exchange DNA material similar to the actions in bacterial recombination. Specialized transduction. Specialized transduction occurs when a prophage is excised in the lysogenic cycle. In this process, by which the restricted set of bacterial genes is transferred to another bacterium, these genes that are located adjacent to the prophage are transferred due to improper excision. Specialized transduction occurs when a prophage excises imprecisely from the chromosome so that the bacterial genes lying adjacent to it included in the excised DNA. The excised DNA along with the viral DNA is then packaged into a new virus particle, which is then delivered to a new bacterium when the phage attacks a new bacterium. Here, the donor genes can be inserted into the recipient's chromosome or remain in the cytoplasm, depending on the nature of the bacteriophage. An example of specialized transduction is the lambda phage in Escherichia coli. Lytic animal viruses undergo stages of infection similar to bacteriophages, including attachment, penetration, biosynthesis, maturation, and release. Although the process of penetration, nucleic acid biosynthesis, and release vary between the two. Animal viruses typically enter host cell via endocytosis or membrane fusion after binding to specific receptors. Many viruses exhibit host specificity, meaning they only infect certain types of host and specific cells within tissues, a characteristic known as tissue tropism. For example, the poliovirus targets the brain and the spinal cord tissues, while influenza virus primarily infects the respiratory tract. Animal viruses exhibit diverse mechanisms for gene expression that do not always adhere to the standard flow of genetic information following the central dogma of molecular biology, which is typically DNA to RNA to protein. While some viruses possess a double-stranded DNA or dsDNA genome and can utilize the normal flow, others have single-stranded DNA, double-stranded RNA, or single-stranded RNA genomes. The type of genome dictates the replication and expression processes for viral proteins. For instance, if a virus has an single-stranded DNA genome, host enzymes synthesize a complementary second strand, resulting in the double-stranded DNA that can undergo replication, transcription, translation, akin to cellular DNA. In contrast, RNA viruses require different mechanisms for gene expression. There are three categories of RNA genomes: double-stranded RNA, dsRNA, positive single-stranded RNA plus ssRNA, the negative single-strand RNA minus ssRNA. Viruses with plus ssRNA genome can be directly translated into the viral protein, functioning similarly to the cellular mRNA. Conversely, for the viruses with minus ssRNA is converted into plus ssRNA by viral enzyme RNA-dependent RNA polymerase or RDRP, which is introduced by the virus itself. This enzyme also plays a critical role in the replication of the double-stranded RNA viruses, using the negative strand of the genomic double helix as a template to generate the plus ssRNA, which can be subsequently translated into the host ribosome. Retroviruses, such as HIV, utilize a unique mechanism for viral nucleic acid synthesis, being classified as plus ssRNA viruses. They contain the enzyme reverse transcriptase within their capsid, which synthesizes a complementary single-stranded DNA or cDNA copy from the ssRNA genome. This cDNA is converted into double-stranded DNA, which can integrate into the host chromosome, forming a provirus that becomes a permanent part of the host's genetic material. This allows the virus to persist within the host, establishing chronic infections, and the provirus stage resembles the prophage stage of the bacterial infection during the lysogenic cycle. Although the provirus does not undergo excision after integration into the genome. We will discuss this process in much more detail beyond the textbook as appropriate for a graduate level course in the following few slides. Please recap from other courses that you all have taken what DNA transcription and DNA replications are. The process of transcription and replication both involve the DNA as the starting material, as every organism have DNA as their genetic material. But viruses, RNA can be the genetic material other than DNA. This is a self-explanatory schematic of the organization of the nucleic acid. Use this diagram to work on your own as a bit of homework, which is not an assignment. If you are given one of the sequence and then asked to write the sequences of the mRNA and the genome replication sequence, you should be able to write it. Please note that the replication sequence is easy. It is the exact copy of what you are seeing and needs no brain at all. For the mRNA, the sequence will be 5' to 3' plus strand sequence of the genome, where the T's or the thymine nucleotides have been replaced by the U's or the uracil nucleotides. It would help if you also practice deducing the complementary intermediate stand that must be made to reach the target sequence of mRNA sequence. Please note that in some viruses, the genome can be an mRNA. Also refer to the notes of slide number 25. Please use this slide along with the slides with notes number 25, 26, 27, and 28 to understand and grasp the two concepts: genome replication, mRNA synthesis, also known as transcription. Please note that the slide number 17, Baltimore scheme, the genome replication was not shown. It only shows the transcription or mRNA synthesis from the genome, whatever it may be. The scheme in this slide shows how different enzymes replicate the viral genomes and transcribe them into mRNAs. These replication and transcription is carried out by certain polynucleotide polymerases of either host or viral origin, and they are named in a very specific way as the following. A. What is the template from which DNA or RNA will be made? If you answer it's DNA, then the enzyme will be DNA dependent. If your answer is RNA, then the enzyme will be RNA dependent. B. What is the enzyme making or polymerizing? If the enzyme is polymerizing DNA, then it will be a DNA polymerase. If the enzyme is polymerizing RNA, it will be a RNA polymerase. More important facts, the DNA-dependent DNA polymerase, DP, is a replication enzyme and can be the host, the cell the virus is infecting, or viral origin. This makes DNA to DNA. DNA-dependent RNA polymerase, DDRP, is a transcription enzyme and can be of host, the cell the virus is infecting, or the viral origin. This makes DNA to RNA. RNA-dependent RNA polymerase, RDRP, is always viral origin. The same enzyme do both replication and transcription. That means it takes mRNAs as well as genome RNAs. Each type of virus within the 7 Baltimore classes has its own unique life cycle. While they generally proceed through the steps: attachment, entry, penetration, biosynthesis, assembly and maturation, 5 release, there are significant differences in how they execute these steps. Consider it like learning about a car engine representing an animal viral life cycle and being shown the working of a Toyota Camry V4 engine, for instance, HIV. In comparison, there were engines as simple as a 50cc single-cylinder engine of the 1885 Benz motor wagon, up to the sophisticated W16 or V16 engines of a Bugatti Chiron or Tourbillon. Understanding the Camry engine, the HIV life cycle, may provide some insight into the concept of an internal combustion engine, but it only scratches the surface of the extensive diversity that exists. However, this discussion is beyond the scope of this introductory microbiology course. So let's take a look at one animal virus life cycle, the life cycle of HIV from entry to exit in a host cell. Human immunodeficiency virus is a retrovirus that specifically targets the immune system, particularly the CD4 plus T lymphocytes. Understanding the life cycle of HIV is crucial for devising therapeutic interventions and vaccines. The life cycle can be divided into several key stages: attachment, entry, biosynthesis, which includes reverse transcription, integration, transcription translation. Number 4, assembly, and the number 5 is release. It is called budding in the enveloped viruses. And number 6, maturation. Entry and fusion. HIV enters the host cell primarily through the interaction between the viral envelope glycoprotein GP20 and CD4 receptor present on the surface of the CD4 plus T cells. Upon binding to CD4, GP120 undergoes a conformational change, allowing it to interact with the co-receptor either CCR5 or CXCR4, depending on the viral strain. This interaction facilitates the fusion of the viral envelope with the host cell membrane, a process mediated by GP41, another viral glycoprotein. Once the fusion occurs, the viral capsid is released into the cytoplasm of the host cell. Reverse transcription. Inside the cytoplasm, the viral RNA genome is reverse transcribed into double-stranded DNA by the viral enzyme reverse transcriptase, or RT. This is carried by the virus. The enzyme possesses both RNA-dependent DNA polymerase and ribonuclease H activities, enabling it to synthesize complementary DNA, cDNA, from the viral RNA template, while simultaneously degrading the RNA strand. The resultant double-stranded viral DNA is now complexed with the viral proteins, forming a pre-integration complex. Integration. The pre-integration complex is transported into the host cell nucleus, where the viral DNA is integrated into the host genomic DNA by the viral integrase enzymes. This integration is a critical step as it allows the HIV to hijack host cellular machinery for its replication. The integrated viral DNA, now referred to as a proviral DNA, remains dormant in the host genome until activated. Transcription and translation. Upon activation, the proviral DNA is transcribed into viral RNA by the host RNA polymerase II. This RNA serves dual purposes. It is used as the genomic RNA for new virions and as the messenger RNA mRNA for the translation of viral proteins. Some viral genes are expressed as polyproteins, which are subsequently cleaved by the viral protease into functional proteins, including structural proteins like GAG, Pol, envelope, the regulatory proteins like TAT, REV, or NEF. Assembly and budding. The assembly of the new virions occurs at the host cell membrane. The structural proteins, along with the newly synthesized RNA genome, are gathered at the inner surface of the plasma membrane, where they form immature viral particles. The viral envelope is acquired from the host cell membrane during this process. Budding is the final step, wherein the mature virions is released from the host cells through an exocytosis-like mechanism. This process, facilitated by the interaction of the viral protein GAG with the plasma membrane and further enhanced by cellular factors. Maturation. Note, if many enveloped viruses, maturation happens after assembly before release. Here it happens after release or budding. All are different. After budding, the immature virion undergoes a maturation process in which the viral protease cleaves the polyproteins, resulting in the structural rearrangement of the core. This maturation is essential for the virion to acquire its infectious capability, enabling it to infect additional CD4+ T cells. Acute infections are caused by viruses that invade the body and replicate quickly, leading to a range of symptoms that can vary in severity. These infections typically manifest suddenly and can affect various systems within the body, such as the respiratory, gastrointestinal, or central nervous system. Common examples include influenza, the common cold, COVID-19. Symptoms may include fever, fatigue, cough, body aches, and in some cases, gastrointestinal distress. While many viral acute infections resolve on their own with rest and hydration a week or two, some may require medical attention, especially in a vulnerable population such as the elderly or those with compromised immune systems. Persistent infection occurs when a virus remains in the host body without being completely cleared, residing in specific tissues or organs. This virus may remain dormant or cause mild infections without significantly harming the host. Factors contributing to the persistent infection can include the regulation of a viral or host gene expression and changes to the host's immune system. There are two main types of persistent infection, latent and chronic. Examples of latent infections include herpes simplex virus, varicella zoster virus, Epstein-Barr virus, while hepatitis C virus and HIV are notable examples of chronic infections. Persistent latent infection. Not all animal viruses replicate through the lytic cycle. Some can remain dormant inside a cell in a process called latency, leading to latent infections. Latent viruses may cause an acute infection initially, such as the varicella zoster virus. It's a herpes virus, which leads to chickenpox characterized by blistering rash. After 10 to 12 days, the infection resolves, and the virus becomes dormant within the nerve cell ganglia for years without killing cells or replicating. The reasons for this dormancy are unclear, but eventually the virus can reactivate after many years, causing shingles, which is a disease that affects nerve cells specifically, unlike chickenpox, which impacts various body areas supplied by those specific nerve ganglia known as dermatomes. Latent viruses can remain inactive by existing as circular viral genome molecules separate from the host chromosome or by integrating into the host chromosome as proviruses. During this dormant phase, they do not produce symptoms of disease, making them challenging to detect. Consequently, individuals may be unaware of their viral infection unless they undergo a viral diagnostic test. Persistent chronic infection is characterized by symptoms that persist or recur over a long duration, often due to the body's inability to eliminate the virus. HIV is a prime example, remaining detectable in tissues after infection, typically without symptoms for years due to the mechanism that hinder the immune function, such as preventing viral antigen expression or infected cells and altering immune cells. This chronic persistence can eventually damage the immune system, leading to acquired immunodeficiency syndrome or AIDS. Similar evasion strategies are observed in other viruses like hepatitis C, which is also cause chronic infection. Acute infection by varicella zoster manifests as chickenpox. Infection is initiated on the conjunctiva or mucosa of the upper respiratory tract, spreads to regional lymph nodes. After 4 to 6 days, infected T cells enter bloodstream, causing a primary viremia. These infected cells subsequently invade the liver, spleen, and other organs, initiating a second round of infection. Virus particles and infected cells are then released into the bloodstream in a secondary viremia. Infected skin-homing T cells efficiently invade the skin and initiate a third round of infection about 2 weeks after the initial infection. The characteristic vesicular rash of chickenpox appears as a result of immune defensive action. Next, the virus particles produced in the skin infect sensory nerve terminals and spread to dorsal root ganglia of the peripheral nervous system, where a latent infection is established. The latent infection is maintained by active immune surveillance. Later in life, perhaps as the robustness of the immune system wanes, viral reactivation can occur, in which another infectious cycle is initiated. In this case, virus particles leave the peripheral neurons to infect the skin. The characteristic recurrent disease called shingles is often accompanied by a long-lasting painful condition called post-herpetic neuralgia. Normally, an infected individual experiences only one visible reactivation event, probably because reactivation stimulates the immune system. Such restimulation of the immune system is the rationale for administering the varicella zoster vaccine, available as Shingrix, to adults to prevent reactivation and shingles. Infection and latency in the peripheral autonomic ganglia. Viral reproduction occurs at the site of infection, usually in the mucosal epithelium. The infection may or may not manifest as a lesion. The infection also spreads locally between the epithelial cells and may spread to deeper layers to engage fibroblasts, capillary endothelial cells, sweat glands, and other dermal cells such as those present in the piloerector muscles around the hair follicles. Particles that are released from the basal surface infect nerve terminals in close contact. These axon terminals can derive from sensory neurons in dorsal root ganglia, left, or on the autonomic neurons in sympathetic ganglia, right. Viral envelopes fuse with neuron axonal membranes, and the nucleocapsid with outer tegument proteins is transported within the axon to the neuronal cell body by microtubule-based system like dynein motors, where viral DNA is delivered to the neuronal nucleus. Spread of productive infection to the central nervous system from these peripheral nervous system ganglia is rare. Unlike the brain and the spinal cord, peripheral nervous system ganglia are in close contact with the bloodstream and are exposed to lymphocyte and humoral effectors of the immune system, immune surveillance. Consequently, infected ganglia become inflamed and populated with lymphocyte and macrophages. Infection of the ganglia usually resolve within 7 to 14 days after primary infection. Virus particles are cleared and a latent infection of some neurons in the ganglia is established. Epstein-Barr virus primary infection. Epstein-Barr virus, also known as human herpes virus 4, primarily infects epithelial cells in oropharynx, particularly in the tonsils where the initial infection occurs. After infecting these epithelial cells, the virus can target resting B cells with the lymphoid tissues, leading a latent infection. The virus-infected B cells produced various latent viral proteins and RNAs, such as latent membrane protein, LMP1 and LMP2. These proteins influence the behavior of the B cells, affecting the differentiation, migration, and potential for tumorigenicity. The infected B cells are subsequently stimulated to proliferate and produce antibodies. However, they also become targets for the immune system, specifically natural killer cells and cytotoxic T lymphocytes, which aim to eliminate virus-infected cells. Persistent infection of Epstein-Barr virus. Despite the immune response that typically kills the majority of the infected B cells, a small fraction, approximately 1 in 100,000, manage to survive and persist as non-proliferative memory B cells that predominantly express LMP2A mRNA. These memory B cells serve as a long-term reservoir for EBV and are capable of producing infectious virus when cultured outside the body. The immune response to these cells can lead to a variety of outcomes, including self-limiting proliferation, infectious mononucleosis, or even unlimited proliferation, which may result in conditions like polyclonal B-cell lymphoma. When these latently infected B cells are stimulated, they can revert to a productive viral state, producing viral proteins necessary for replication and maintaining the viral genome. Some of these B cells migrate to lymphoid tissues near epithelial cells in the oropharynx, where they can reactivate and shed infectious particles into saliva, facilitating transmission to new hosts. Implication of Epstein-Barr virus latency and reactivation. EBV establishes latent infections primarily in B lymphocyte and is associated with several human cancers, highlighting its clinical significance. The mechanism of B cell immortalization during latency does not provide any selected advantage to the virus itself, unlike non-pathogenic latent state of other herpes virus. Initial infections typically occurs in childhood or adolescence, often through oral contact, leading to infectious mononucleosis characterized by a robust immune response against the virus. While most infected cells are eradicated, the surviving memory B cells do not express certain coactivator receptors, allowing them to evade cytotoxic T lymphocyte detection and become immortalized. These immortalized B cells synthesize a variety of viral protein, including nuclear proteins and small RNAs, but the roles of many of these proteins in cancer development remains poorly understood. Reactivation of EBV is complex and involves a cascade of signal transduction that activates essential viral transcription factors, ultimately leading to a productive infection and shedding of viral particles. The human immunodeficiency virus HIV are two species of lentivirus that infect humans and lead to acquired immunodeficiency syndrome. Two types of HIV have been identified, HIV-1 and HIV-2. HIV-1, initially known as lymphoadenopathy-associated virus LAV, and the human T lymphotropic virus 3 or HTLV3, is more virulent and infectious than HIV-2, accounting for the majority of the global HIV infection. In contrast, HIV-2 has a lower infectivity, meaning fewer individuals exposed to it will become infected, and is primarily confined to the West Africa due to its limited transmission capability. Chronic infection with human immunodeficiency virus represents a complex interplay between the virus and the host's immune system. Following initial infection, the HIV targets CD4 plus T lymphocyte, leading to their progressive depletion. This depletion is primarily due to the direct viral cytopathy, immune-mediated destruction, and the establishment of viral reservoirs. The acute phase of infection is characterized by high viral loads and the presence of acute retroviral syndrome, ARS, which often leads to robust immune response and the production of neutralizing antibodies. As the infection becomes chronic, the viral load typically stabilizes, entering a phase referred to as clinical latency or chronic HIV infection. During this phase, the virus persists in a quiescent stage within lymphoid tissue, particularly in the gut-associated lymphoid tissue or GALT, where it remains shielded from the both immune surveillance and antiretroviral therapy or ART. This latency is maintained by various mechanisms, including the integration of the viral genome into the host DNA and the establishment of long-lived latency-infected memory T cells. The chronic nature of HIV infection is also marked by immune activation and inflammation, which paradoxically occurs despite the presence of ART. Persistent immune activation is associated with elevated levels of pro-inflammatory cytokines and increased turnover of CD4 plus T cells, contributing to the development of non-AIDS-related comorbidities, such as cardiovascular disease and neurocognitive disorders. Current therapeutic strategies focus on the use of the ART to suppress the viral replication and improve immune function. However, challenges remain in achieving a cure for HIV, primarily due to the existence of viral reservoirs and the potential for rebound viremia upon cessation of therapy. Ongoing research explores novel approaches, including latency-reversing agent and gene-editing technologies, to target and eliminate these reservoirs, offering hope for future advancement in the management of chronic HIV infection. Plant viruses share more similarities with animal viruses than bacteriophages, possessing either an enveloped or non-enveloped structure. They can have a DNA or RNA genome, with most plant viruses being predominantly composed of a positive sense single-stranded RNA plus ssRNA, which functions similarly as messenger RNA or mRNA. While a minority of plant viruses may contain different genome types, the majority of them lack a DNA genome. The host range of plant viruses can be narrow or broad. For instance, Citrus tristeza virus specifically infects certain plants within the citrus genus, while the cucumber mosaic virus can infect a wide variety of plants across numerous families. Transmission of these viruses typically occur through physical contact between the plant or via vectors such as insects, nematodes, fungi, and other arthropods. Some viruses are reliant on specific insect vectors for transmission, meaning they may be spread only by certain insects like aphids, but not by others like whiteflies. Additionally, viruses can enter healthy plants through wounds caused by pruning or environmental damage. Plant viruses are categorized as biotrophic parasites, allowing them to infect hosts without causing immediate death, akin to lysogenic cycles of bacteriophages. Viral infections can be asymptomatic or result in cell death, with the infection process starting when the virus penetrates the host cell. After uncoating, the virus uses the host cell's machinery to replicate its genome, produce proteins for new virion assembly. For a systemic infection to occur, the virus must access the plant's vascular system, such as the phloem, with the time needed for systemic infection varying based on the virus, plant species, and environmental factors. The life cycle concludes when the virus is transmitted from an infected plant to a healthy one. The slide shows a general view of virus cell-to-cell and long-distance movement in plant tissues. After inoculation, mostly into epidermal or mesophyll cells, virions are disassembled for replication and translation of the viral genome. Viral proteins, sometimes associated to cellular factors, interact with the viral genome to form the transport complexes, virions, or RNP, ribonucleoprotein complexes, allowing virus movement from cell to cell via plasmodesmata. Viral replication and cell-to-cell movement continue in and between nucleate phloem cells, example, bundle sheath, vascular parenchyma, and companion cells. Then the transport complexes in form of the virion or RNP are loaded into a sieve element for long-distance movement before being finally released into systemic tissues to start a new infection site. The whole process requires an effective crossing of successive boundaries between different cell types and leading to systemic infection of the plant. The growth curve of virus population throughout its life cycle does not resemble a sigmoidal curve typical of bacterial populations. Initially, a viral inoculum initiates infection. During the eclipse phase, the virus attaches to and enter host cells, but no virions are detectable in the surrounding media. A key difference in the viral growth curve compared to the bacteria becomes evident when virions are simultaneously released from the lysed host cell. This event is referred to as a burst, and the number of virions released per bacterium is known as a burst size. In a one-step multiplication curve for bacteriophages, host cells lyse and release numerous virus particles into the medium, resulting in a rapid increase in the viral titer, the concentration of virions per volume. Once no viable host cells remain, the viral particles start to degrade, leading to decline in the culture's viability. Viruses were initially referred to as filtrable agents due to their ability to pass through filters that could trap bacteria, indicating their unique nature as infectious agents. The cultivation of viruses involves specific methods for specimen collection and handling, ensuring that these delicate entities are preserved and studied effectively. Additionally, there are distinct techniques for cultivating viruses which can be categorized into in vivo methods, where viruses are grown within living organisms, and in vitro techniques where they are cultivated in controlled laboratory environments. Unlike bacteria, which can often be cultivated in artificial nutrient media, virus necessitates a living host cell for replication. Infected host cells, whether eukaryotic or prokaryotic, can be cultured and grown, allowing for the growth media to be harvested as a source of the virus. The virions present in the liquid medium can be separated from the host cells through a process such as centrifugation or filtration. Filters effectively remove any particles larger than the virions from the solution, enabling the collection of the viruses in the filtrate. Viruses can be cultivated either in vivo, which means within a living organism such as a plant or animal, or in vitro outside a living organism in an artificial environment like a test tube, cell culture flask, or agar plate. Bacteriophages, a type of virus that infects the bacteria, can be grown on a dense layer of bacteria known as bacterial lawn, which is cultivated on 0.7% soft agar within a petri dish or flat flask. This concentration is lower than the typical 1.5% used for growing bacteria, allowing the bacteriophages to diffuse easily through the medium. For lytic bacteriophages, the lysis of the bacterial host becomes observable when a clear zone called plaques appear in the bacterial lawn. As the phages kill the bacteria, numerous plaques can be seen amidst the cloudy bacterial culture. Animal viruses need to infect cells from host animal or tissue cultured cells that come from that animal. Cultivating animal viruses is crucial for several reasons. First, it helps in identifying and diagnosing harmful viruses in clinical samples. Second, it is essential for producing vaccines. And third, it supports basic research studies. In vivo source for these viruses can include developing embryos in bird cages like those of chickens or turkeys or entire animal. For instance, a significant amount of influenza vaccine produced for yearly flu vaccine programs is grown in hens' eggs. The embryo or host animal acts as incubator for the replication of the virus. The specific location within the embryo of a host cell is vital, as many viruses have tissue tropism, meaning they need to be introduced into a particular site for optimal growth. In embryos, these target sites may include the amniotic cavity, the chorioallantoic membrane, or the yolk sac. Viral infections can damage tissue membranes, leading to lesions known as spocks, disrupt the development of the embryo, or even result in the embryo's death. In vitro studies and cell culture. In vitro studies utilize various types of cell supports virus growth. A primary cell culture is created from a freshly obtained animal organs or tissues. Cells can be extracted through mechanical scraping or minching, or release them by using enzymatic methods like trypsin or collagenase to break down the tissue. Since primary cell culture requires a solid surface for attachment and growth, they are maintained in a liquid culture medium within a petri dish or tissue culture flask. These cultures typically have a limited lifespan. As cells undergo mitosis and reach certain density, they contact each other, which triggers a process called contact inhibition, stopping further mitosis to prevent overcrowding. To avoid contact inhibition, cells must be moved to a new vessel with fresh growth medium, resulting in a secondary cell culture. Additionally, to maintain cell growth, the density of the cell must be managed by periodically removing some cells and adding fresh media. Immortal cell lines, the HeLa example. An example of an immortal cell line is the HeLa cell line, originally cultivated from tumor cells taken from Henrietta Lacks, a patient who passed away from cervical cancer in 1951 at Johns Hopkins University Hospital. HeLa cells were the first continuous tissue culture cell lines and played a crucial role in establishing tissue culture as a fundamental technology in cell biology, virology, and medicine. Before the advent of HeLa cells, scientists struggled to create reliable tissue cultures. Over 6 decades later, this cell line continues to be used in medical research. For more information about this significant cell line ethical controversy surrounding its acquisition, see the Eyes on Ethics, the immortal cell line of Henrietta Lacks. Once a virus is introduced into the host organism, embryo, or tissue culture cell, a sample can be prepared for further analysis using bright field electron or fluorescent microscopy. Cytopathic effects, CPE, are distinct observable abnormalities in cells caused by viral infection, which may include loss of adherence to the container surface, changes in the cell shape from flat to round, shrinkage of the nucleus, presence of vacuoles in the cytoplasm, fusion of cytoplasmic membranes, formation of multinucleated cells called syncytia, and inclusion bodies in the nucleus or cytoplasm, culminating in the complete cell lysis. Additionally, viral infection can lead to disruption of the host genome and transformation of normal cells into transformed cells, which are linked to carcinomas and sarcomas. The type and severity of CPEs vary based on virus involved. Hemagglutination assay. A serological assay is the test that helps find certain types of viruses in a patient's serum, which is the surface liquid part of the blood plasma after the clotting factors have been removed. This serum can be used in a direct test known as a hemagglutination assay to identify specific viruses in the sample. Hemagglutination refers to the clumping of the red blood cells or erythrocytes. Some viruses have proteins on their surface called hemagglutinins that can attach to receptors on the red blood cells, causing them to clump together. This clumping can be seen without a microscope. However, the method may not clearly distinguish between infectious and non-infectious viral particle, as both can cause agglutination of the red blood cells. To pinpoint a specific pathogenic virus using hemagglutination, an indirect method is required. Antibodies, which are proteins produced by the patient's immune system to combat specific viruses, can bind to the hemagglutinins associated with these viruses. When antibodies attach to the hemagglutinins on the virus, they prevent red blood cells from interacting with the virus directly. As a result, when red blood cells are introduced to the virus covered in antibodies, agglutination does not occur. This means agglutination has been inhibited. These indirect assays for virus-specific antibodies are known as hemagglutination inhibition assay. HAI can detect the presence of antibodies for various viruses that may cause an infection even months or years after the initial infections. In Figure 2, in Panel A, displays a control experiment featuring several viral dilutions. In the absence of hemagglutinins, erythrocytes settle at the bottom, creating a button appearance. Conversely, figure 2 panel B illustrates red blood cells mixed with various virus dilutions. The virus is detectable up to 1:512th dilution. Beyond this dilution, the viral presence is too low to induce hemagglutination, causing the erythrocytes to sink at the bottom of the well. Notably, the 1:4 dilution also exhibits a button. A very high concentration, the viral enzyme neuraminidase on the membrane cleaves the glycosylation or hemagglutinins, preventing them from functioning as hemagglutination agents. Neuraminidase enzymes are present in minimal amount on the viral surface, allowing them to cleave hemagglutinins glycosylation only at a certain concentrated viral titer. Nucleic acid amplification tests, NAT, are important tools in molecular biology for identifying specific nucleic acid sequences of viruses and in patient samples. One common method is the polymerase chain reaction, or PCR, which helps to find the viral DNA in tissues or fluids from patients. PCR works by creating many copies of particular viral DNA segment. This is done using a short sequence called primers that attach to specific parts of the viral DNA, allowing for the identification of the virus. The PCR methodology involves a series of precise steps that are repeated for several cycles to exponentially increase the quantity of the target DNA. The process begins with denaturation phase, where the reaction mixture containing the DNA sample, primers, nucleotide, and heat-stable DNA polymerase enzyme is heated around 94 to 98 degrees Celsius. This high temperature causes the double-stranded DNA to separate into 2 single strands. Next, the temperature is lowered by approximately 50 to 65 degrees Celsius during the annealing phase, allowing the primers, short sequences of nucleotides that are complementary to the target DNA region, to bind to their specific sites on the single-stranded DNA template. This is the most crucial step, as the specificity of the primers determines the accuracy of the amplification. Following annealing, the temperature is raised to about 72 degrees Celsius for the extension phase, where the DNA polymerase synthesizes new DNA strands by adding nucleotides to the 3' end of the primers, effectively creating a new double-stranded DNA molecule. These 3 steps, denaturation, annealing, and extension, constitute one cycle of PCR, and this cycle is repeated 25 to 35 times. As a result, a single copy of a target DNA can be amplified into millions of copies, making it easier to analyze and manipulate. The entire PCR process is highly sensitive, allowing for the detection and amplification of DNA from minute samples, which is not only useful in detecting viruses in their samples, but also useful in applications such as genetic testing, forensic analysis, and other infectious disease diagnosis. Reverse transcription polymerase chain reaction, or RT-PCR, is a variation of the standard PCR technique that is specifically designed to amplify RNA sequences. The methodology begins with the extraction of RNA from the sample, but if the infectious virus is an RNA virus, a direct patient sample would contain viral RNA, which is then reverse transcribed into complementary DNA, or cDNA, using the enzyme reverse transcriptase. This cDNA serves as a template for the PCR amplification process. Following the reverse transcription step, the cDNA undergoes the same PCR process, which includes denaturation, annealing, and extension. This allows researchers to quantify gene expression levels by amplifying specific RNA sequences, making RT-PCR a valuable tool in studying gene regulation, viral infections, and another RNA-related research. Quantitative polymerase chain reaction, qPCR, also known as real-time PCR, is a sophisticated technique that allows for the quantification of specific DNA or RNA sequences during the amplification process. In qPCR, the amplified DNA is monitored in real time through the use of fluorescent dyes or probes that emit fluorescence when bound to double-stranded DNA. This fluorescence can be measured at each cycle of the PCR, providing a continuous assessment of the reaction's progress. One of the key matrices in qPCR is the threshold cycle, or CT value, which is defined as the number of cycles required for the fluorescence to surpass a defined threshold level. The lower the CT value, the higher is the initial quantity of target nucleic acid present in the sample, as fewer cycles are needed to detect the signal. This quantification ability makes qPCR particularly valuable in clinical diagnosis, including the detection and measurement of viral infections. For instance, when diagnosing for a viral infection such as SARS-CoV-2, samples from patients, often in the form of respiratory swabs, are analyzed using qPCR to detect the presence of viral RNA. By comparing CT values from patient samples to those from known standards or controls, clinicians can determine not only the presence of the virus, but also the viral load, which is critical for understanding the severity of the infection and guiding treatment decisions. Thus, qPCR serves as a vital tool in virology, enabling rapid and accurate assessment of viral infections, monitoring disease progression, and evaluating the effectiveness of therapeutic interventions. Enzyme immunoassay (EIA) is a generic name, and the most common form is the enzyme-linked immunosorbent assay, or ELISA. ELISA is a highly sensitive and versatile laboratory technique used to detect and quantify proteins, antibodies, and antigens, making it an essential tool in the diagnosis of viral infections. The ELISA method involves immobilizing an antigen, the virus or viral protein, onto a solid surface, typically a microplate, and then adding a sample that may contain antibodies specific to that antigen. If antibodies are present in the sample, they will bind to the immobilized antigen, forming an antigen-antibody complex. Following this, an enzyme-linked secondary antibody that recognizes and bound antibodies is added, creating a sandwich complex. After washing away unbound antibodies, a substrate for the enzyme is introduced, and the color change occurs as the enzyme catalyzes the reaction, producing a measurable signal. The intensity of color is directly proportional to the amount of antigen present in the sample, allowing for quantitative assessment. ELISA is particularly advantageous for virus detection because it can be tailored to identify specific viral proteins or antibodies produced in response to a viral infection. For example, in the diagnosis of diseases like HIV, hepatitis B, or influenza, ELISA can be deployed to detect either the viral antigen in the patient sample or antibodies that indicate prior exposure to the virus. This technique not only provides a rapid result, but also allows for high-throughput testing, making it a crucial tool in clinical settings and epidemiological studies to monitor outbreaks and assess immune response in populations. Viroids, virusoids, and prions. Research into the causes of previously unstudied diseases has uncovered non-living disease agents that are significantly different from viruses. These agents include particles made solely of RNA or protein, which can still replicate by taking over a host, similar to viruses, enabling them to cause disease. So far, these findings have identified viroids, virusoids, and proteinaceous prions. Viroids, virusoids, and prions are distinct infectious agents that exhibit unique characteristics. Viroids are small circular RNA molecules that lack a protein coat and are known to cause diseases in the plants. Virusoids, on the other hand, are similar to viroids but require a helper virus for their replication and can affect both plants and animals. Prions are misfolded proteins that can undergo abnormal folding of normal proteins in the brain, leading to neurodegenerative disease. Each of these agents has specific structural and functional properties that differentiate them from the traditional viruses. In 1971, Theodore Diner, a pathologist at the Agricultural Research Service, discovered an acellular particle he named a viroid, which means virus-like. Viroids consist solely of a short strand of circular RNA that can replicate itself. The first viroid identified was found to cause potato tuber spindle disease, leading to slower sprouting and various deformities in the potato plants. Similar to viruses, potato spindle tuber viroids, PSTVs, hijack the host machinery to replicate their RNA genome. However, Unlike viruses, viroids lack a protein coat to protect their genetic material. Viroids can lead to significant losses in commercially important agricultural food crops cultivated in the field and orchards. Since the discovery of PSTV, additional viroids have been identified that causes disease in various plants. For instance, tomato planta macho viroid infect tomato plants, resulting in a loss of chlorophyll, deformed and brittle leaves, and small tomatoes, which decreases productivity in the crop. Avocado sunblotch viroid leads to lower yield and fruit of inferior quality, and it is the smallest viroid discovered so far that infects plants. Peach latent mosaic viroid can cause necrosis of flower buds and branches, as well as damage to ripened fruit, which can promote fungal and bacterial growth. PLMVDs also affects plums, nectarines, apricot, and cherries, resulting in reduced productivity in these orchards. Generally, viroids can be spread mechanically during crop maintenance and harvesting through vegetative propagation and possibly via seeds and insects, leading to significant decline in the food availability and severe economic consequences. A second type of pathogenic RNA that can infect commercially important agricultural crops is virusoids, which are subviral particles best described as non-self-replicating single-stranded RNAs. The RNA replication of the virusoids is similar to that of viroids. However, unlike viroids, virusoids require the cell to also be infected with a specific helper virus. Currently, there are only 5 described types of virusoids and their associated helper virus, all of which belong to the Sobemovirus family. An example of a helper virus is the subterranean clover mottle virus, which packages an associated virusoid into its viral capsid. Once the helper virus enters into the host cell, the virusoids are released and can be found free in the plant cell cytoplasm, where they exhibit ribozyme activity. The helper virus replicates independently of the virusoids until the virusoid genomes are relatively small, ranging from 220 to 388 nucleotides in length. Importantly, a virusoid genome does not code for any protein. It solely serves to replicate the virusoid RNA. Virusoids are part of a larger group of infectious agents known as satellite RNAs, which are similar to pathogenic RNAs found in animals. Unlike plant virusoids, satellite RNAs can encode proteins. However, like plant virusoids, they must also co-infect with the helper virus to replicate. One satellite RNA that infects humans and has been described by some scientists as a virusoid is the hepatitis delta virus, which is also referred to as hepatitis delta virusoid. HDV is much larger than a plant virusoid, possessing a circular single-stranded RNA genome of 1700 nucleotides, which allow it to direct the biosynthesis of HDV-associated proteins. The helper virus for HDV is the hepatitis B virus, or HBV. Co-infection with HBV and HDV leads to more severe pathological changes in the liver during infection, which is how HDV was first discovered. At one time, scientists believed that any infectious particle must contain DNA or RNA. However, in 1982, Stanley Prusiner, a medical doctor studying scrapie, a fatal degenerative disease in sheep, discovered that the disease was caused by proteinaceous infectious particles known as prions. Since proteins are acellular and do not contain DNA or RNA, Prusiner's finding faced initial resistance and skepticism. Nonetheless, his research was ultimately validated, leading him to receiving Nobel Prize in Physiology and Medicine in 1997. A prion is a misfolded version of normal protein PRPC found within the cells. This normal prion protein PRPSC can arise due to the genetic mutations or occur spontaneously and has the potential to be infectious, causing other normal proteins to misfold as well, which leads to formation of plaques. Prions are known to cause various types of transmissible spongiform encephalopathy, or TSE, in both humans and animals. TSE is a rare degenerative disorder that impacts the brain and the nervous system. The buildup of these rogue proteins result in the brain tissue becoming sponge-like, which destroyed brain cells and create holes in the tissue, resulting in brain damage, loss of motor coordination, and dementia. Infected individuals experience mental impairment and may become unable to move or communicate. There is no cure for TSC, and the disease progresses quickly, ultimately leading to death within a few months to years. Prions are highly resilient agents and are challenging to eliminate due to their resistance to heat, chemicals, and radiation. Standard sterilization methods often fail to destroy these particles effectively. At present, there is no treatment or cure for transmissible spongiform encephalopathies or TSC diseases, necessitating strict adherence to the federal guidelines for handling contaminated meat or infected animals to prevent transmission. The prion protein is a unique type of protein that can exist in two distinct conformations, the normal form PRPC and the scrapie form PRPSC. Normal prion protein PRPC is a glycophosphatidylinositol or GPI-anchored protein that is predominantly found on the surface of the neurons and other cell types. In its normal state, PRPC is characterized by high proportion of alpha helical structure, which are believed to play a role in cellular signaling and neuroprotection. This conformation is soluble and non-toxic to cell. It is thought to have a physiological role in cellular processes, including cell adhesion and protection from oxidative stress. In contrast, scrapie prion protein PRPCSC is the misfolded and infectious form of the prion protein associated with the prion disease, such as scrapie in sheep, Creutzfeldt-Jakob disease in humans, bovine spongiform encephalopathy or BSC, mad cow disease in cattle. PRPSC is characterized by a higher content of beta sheet structures, which render it insoluble and resistant to proteolytic degradation. This misfolding leads to accumulation of PRPSC in neural tissues, causing neurodegeneration and spongiform changes in the brain. PRPSC has the ability to induce misfolding of normal PRPC into more PRPSC, perpetuating a cycle of infection and leading to the clinical manifestation of prion disease, which include severe neurological symptoms and ultimately death. The key difference between these two forms lie in their conformation, solubility, and biological activity. While PRPC is benign and involved in a normal cellular function, PRPSC is pathogenic, leading to devastating neurological disorders through its ability to propagate and accumulate in the brain. The understanding of these differences is crucial for the developing of diagnostic tools and therapeutic strategies for prion disease. The figure shows how the beta sheets cause the proteins to form this amyloid or the fibrous core. The fibrous structure destroys the cell integrity by killing the neuron where it happens. That neuron is never replaced as they are amyloidic and do not divide by cell division. Transmissible spongiform encephalopathies TSCs in humans include Kuru, fatal familial insomnia, Gerstmann-Straussler-Scheinker's disease, and Creutzfeldt-Jakob disease. In animal TSC, such as the mad cow disease, scrapie found in sheep and goats, and chronic wasting disease in elk and deer are prevalent. These diseases can be transmitted between animals and from animals to humans through consumption of contaminated meat or animal feed. Human transmission may occur through hereditary, particularly in cases of Gerstmann-Straussler-Scheinker syndrome and Creutzfeldt-Jakob disease, or via contact by contaminated tissues such as during blood transfusion or organ transplant. There is no evidence to suggest that TSCs can be transmitted through casual contact with an infected individual. For further details, refer to the table listing TSCs that affect humans and their mode of transmission. Clinical focus: rabies. David, a 45-year-old journalist, returned to the US after traveling to Russia, China, and Africa. Upon his return, he visited his general practitioner due to feeling unwell, presenting symptoms such as weakness in his arms and legs, fever, headache, agitation, and discomfort. He expressed concern that these symptoms might be linked to a dog bite he received while interviewing a farmer in China. Although the dog appeared healthy, David has developed prickling and itching sensation at the bite sites. The doctor conducted a culture and sensitivity test which returned negative for pathogenic bacteria, prompting consideration of further testing for other potential infections. The doctor suspected that David's symptoms might indicate early signs of rabies given the unusual sensation at the skin site of the bite. To confirm or rule out this diagnosis, the physician collected samples of David's blood, saliva, and skin. The skin sample taken from the bite site underwent immunofluorescence staining to detect rabies antibodies in the cutaneous nerves. Additionally, a serum sample was tested for rabies virus antibodies, while the saliva sample was analyzed using reverse transcriptase polymerase chain reaction or RT-PCR to identify viral nucleic acid. The blood test ultimately revealed the presence of rabies virus antigen, leading to a prescription for prophylactic treatment. In conjunction with the RT-PCR analysis, David's saliva underwent viral cultivation to check for active viral presence. However, the result was negative for the virus cultivation, which the doctor indicated was not unexpected, as a single negative test does not definitely rule out rabies. The timing of the test and the quantity of the virus present could affect the results. The doctor considers repeating the test at intervals to improve the chance of detecting any viral presence if it was low during the initial sampling. As day passed, the doctor receiving results from the immunofluorescent test on the skin sample, which shows no rabies antigen, along with another negative test on the saliva sample. Despite these negative results, the doctor opted to continue with David's treatment regime based on the positive RT-PCR outcome. David received an injection of rabies immunoglobulin near the bite site to neutralize any potential rabies virus in his tissues, followed by a series of 4 rabies-specific vaccination over the next 2 weeks to bolster his immune response. Fortunately, David's conditions improved. He made a full recovery thanks to the timely treatment. However, the case highlights the serious nature of rabies, which is almost always fatal once the symptoms appear. Prophylactic treatment are crucial and primary focus on preventing the disease from progressing, as post-bite treatments are mainly palliative in cases where the symptoms have already developed. Rabies viruses have an incubation time of minimum 3 weeks to months. So this is one of those, probably the only kind of almost always fatal viral disease that could be prevented by a post-bite or post-viral exposure vaccine. In the past, patients exhibiting severe symptoms were often restrained and placed in dark rooms, leading to their ultimate fate in a couple of days. Today, when patients develop such symptoms, doctors attempt to treat them with passive antibodies or immunoglobulins against the virus. However, these large molecules often cannot pass the blood-brain barrier where the virus replicates. In these dire situations, patients are sedated to alleviate pain and trauma, typically resulting in death within 2 to 3 days. Although there is an annual death of 60 to 70,000 individuals worldwide, the United States has experienced a significant decline in the human rabies infections and death, primarily due to the vaccination efforts, public education, and prompt post-exposure treatment, with only 1 to 3 cases reported annually. The CDC noted that there were no human rabies cases in 2019 and 2020. 5 deaths occurred in 2021, the highest in a decade, likely due to the decreased awareness of rabies risk. A California art teacher died in November 2024 after a bat bite. An additional rabies-related rates were reported in Minnesota and Kentucky last year. The rise of superbug or multi-drug-resistant bacteria presents a significant challenge for the pharmaceutical industry and poses a serious public health threat. According to 2013 report by the U.S. Centers for Disease Control and Prevention (CDC), over 2 million individuals in the United States are infected with drug-resistant bacteria each year, leading to at least 23,000 deaths. The ongoing use and overprescription of antibiotics are likely to exacerbate the problem by facilitating the evolution of additional drug-resistant strains. One promising solution to combat this issue is a phage therapy, which utilizes bacteriophages. viruses that targets and kill specific bacteria. While phage therapy was first employed in Europe in 1915 by English bacteriologist Frederick Tward, the development of antibiotics led to a decline in its use, except for the former Soviet Union and select Eastern European countries. In recent years, interest in phage therapy has resurfaced due to the increase in antibiotic-resistant bacteria, marking a potential revival of this treatment. Phage therapy offers distinctive advantages over traditional antibiotics. Unlike antibiotics that indiscriminately kill both harmful and beneficial bacteria, phages are specific to particular bacterial strains, preserving the normal microbiota. Additionally, the development of new antibiotics is often costly for pharmaceutical companies and patients, particularly in low-income countries, making phage therapy an attractive alternative. Phage have also been employed to prevent food spoilage. In 2006, the U.S. Food and Drug Administration approved the solution containing 6 bacteriophages that can be applied to lunch meat like bologna, ham, turkey, and eliminate Listeria monocytogenes, a bacterium responsible for listeriosis, a foodborne illness. However, some consumers express concerns about the application of phage in food products, especially with the rising trend of organic foods. To address these concerns, food treated with phage must indicate bacteriophage preparation, ingredients listing, or include labels stating that the meat has been treated with antimicrobial solution to reduce microorganisms. On September 24, 2014, Thomas Eric Duncan arrived at the Texas Health Presbyterian Hospital in Dallas with symptoms resembling a cold or flu, but was misdiagnosed with sinusitis and sent home with antibiotics. 2 days later, his condition worsened and tests confirmed he is infected with Ebola virus, which he contracted after helping a neighbor in Liberia, a country experiencing an Ebola epidemic. Duncan's case highlighted the Ebola virus's incubation period of 2 to 21 days, during which he could not transmit the virus until the symptoms appeared, making the disease highly contagious thereafter. Following his admission, public health officials traced 10 high-risk and 50 low-risk individuals he had contact with, none of whom contracted the disease, although one nurse caring for Duncan did become infected. This situation underscored the necessity for enhanced training and healthcare professionals in the U.S. hospitals to manage emerging epidemics like Ebola and raised questions about the differences between contagious and infectious pathogens, emphasizing the importance of preparedness in the face of public health threats. Eyes on Ethics, Immortal Cell Line of Henrietta Lacks, or HeLa. In January 1951, Henrietta Lacks, a 30-year-old African-American woman from Baltimore, was diagnosed with cervical cancer at the Johns Hopkins Hospital, caused by human papillomavirus, or HPV, which transformed her cells to divide continuously, leading to her death at age 31. Before her passing, samples of her cancerous cells were taken without her knowledge or consent, ultimately leading to the creation of the immortal HeLa cell line by Dr. George Gay, which has since been crucial in various research fields including polio, cancer, and AIDS. Henrietta's family remained unaware of this ongoing use of her cells for over 2 decades, and although the HeLa cells have been commercialized, her estate has never been benefited financially. However, in 2013, they were granted control over the publication of her cell's genetic sequence. The situation raises significant bioethical concerns regarding informed consent, as there were no existing laws or guidelines at that time. While today's standards deem the harvesting of tissues without consent unethical and illegal, the ongoing use of lax cells possess questions about whether the benefits to science justify the means by which these cells were obtained. The ethical implications extend to whether Lacks would have appreciated her contribution to medical advancement and the well-being of countless individuals, or felt violated by the exploitation of her body without consent, leaving her family's wishes and feelings uncertain and unanswered. if you have a chance, please read the book by Rebecca Skloot, The Immortal Life of Henrietta Lacks. These are your links to learning, educator's resource. There are some cool animations and some informatory pages. Thank you.